Good afternoon. This is the 19th of October, 2019, and you have arrived at Authentic Biochemistry and VerEvMed Studios in the Pacific Northwest, where I'm Dr. Dan Guerra, and I'm going to discuss with you today a couple of papers from the primary scientific literature. Today we're going to focus on a topic that I have discussed in the past several times, and it has to do with the neurobiochemistry and neuroimmunoendocrinology of neuropsychiatric disease. And so what we want to do basically is get started with an introduction into membrane action potentials, and then from there move on to our actual papers. So let's get started. All right. So there's the VerEvMed website, and also where you can find the Facebook page where you can get all the necessary information you want to. There's also a YouTube channel, which is where this will end up, this particular video lecture. There's my podcast, Authentic Biochemistry, which is still in audio, and I'm going to be actually migrating my VerEvMed with my Authentic Biochemistry, so I'm going to be able to move back and forth between audio and video in both platforms. I'm Dr. Daniel J. Guerra, and those are the two companies I run, VerEvMed and Authentic Biochemistry, and there is the Facebook page down there. And as I said, today we're going to talk about new neuroimmunoendocrine factors, and we're going to link that up with major depressive disorder. All right, so let's get started. Now, this is a scan, an MRI scan, showing you mild to very severe hyperintensity malformation of the white matter in the brain. So a total burden of white matter disease varies significantly among asymptomatic adults. This would be, for example, in multiple sclerosis, with patients with known cerebrovascular disease, such as MS. In age-matched individuals, white matter hyperintensity volume may vary also between mild to very severe. That's the upper panel, okay? So using validated, semi-automated, what's called a volumetric protocol, which I'm going to get into a little bit when we talk about the paper, the white matter hyperintensity volume can be quantified. And this is really necessary in biochemistry and in biophysics. If you can't quantify something in science, then all you have is a qualitative change or a qualitative difference. And if you can't measure that quantitatively, it's difficult to assign a specific biological conduit or foundation for it. So we want to be able to quantify, and that's what MRI allows us to do. So this particular one used something called supratentorial axial T2 flare, and these are MRI slices. And I've got this from a ResearchGate profile from Arumua Chetunet. All right. Now, in order for you to understand what a white track is in the brain, you have to know that it looks white because you're talking about myelinated axon fibers. Now, I can't talk about, although I'd love to, all about sphingomyelin biosynthesis and how the sphingolipids coat those myelinated axons. And that's why they look that way, because they're basically coated with an insulation of a very complex phospholipid known as sphingolipid. They're not glycerolipids. They contain sphingosine as their backbone, which is basically a condensation of the amino acid serine and the fatty acid palmitate. And then after the introduction of a trans double bond, you make that sphingosine base. Then you start making amide linkages to fatty acids. And then after that, you can add carbohydrate of various structures and complexities of bonding angle. And you make all the different sphingolipids that make up as composition and also in sequence what is then called the myelin sheath. So again, that's all I'm going to say about the lipid part of it. In previous talks, I've gone through that in some detail, and I'll go back to it again. Don't worry, because I'm a lipid biochemist, and it's all about lipids, let me tell you. So when we talk about membranes, here we are, we're going to talk about this paper that's going to suggest that anisotropy in white matter tracts, okay, so that means anisotropy just simply means different morphological shapes or conformations, okay, between a healthy and a non-healthy. is somehow going to be mapped onto a neuropsychiatric disease. Today we're going to talk about major depressive disorder and also schizophrenia, and also bipolar disorder, but there are lots of other such things such as brain damage or brain lesions, or I mentioned before, multiple sclerosis. So it's not just neuropsychiatric diseases that people look at white tracks, that is myelinated axons. But in order to understand any of that, you have to know there's a membrane potential, which follows normal neuro and, in fact, biochemical and biophysical constant change. So the Nernst equation, where in this particular description shown here, you've got delta psi equals RT divided by ZF, natural log of M taken to the Z power of outside divided by the M taken to the Z power inside. Now, what do all of these particular components mean? M to the Z just means an ion of charge Z, a mass of an ion charge Z. So that could be, usually it's a metal, right? Doesn't have to be. In fact, often it's something like sodium or potassium. But when you talk about in half cell chemistry, often it's something like copper or iron. Those of you who have taken organic chemistry recently know what I'm talking about. So the delta psi is actually the membrane potential, and that's measured in volts. The Z is the charge of the ion, as I just mentioned to you. The F is a Faraday constant, and the R is actually the universal gas constant, and that's in joule per degree Kelvin per mole, okay? Reciprocal mole, reciprocal degree Kelvin. The Goldman equation, which is actually more used in the biophysics and biochemistry of action potentials in something like the central nervous system, has to take into account multiple ions. And so you just blow out this equation to include multiple ions, either inside or outside that membrane, and you have to make a summation. It's basically the only difference between the Goldman equation and the old Nernst equation. You're looking at electrochemical potentials across a barrier, in this case, a biological membrane. All right. Now, real quick, speed dial about action potentials. So the action potential has to do with the conductance of two ions. Both of them are cations, sodium, and potassium is the other. So you have a resting state, which is about at minus 61 millivolt. Then you have a hyperpolarization, which goes up to about 55 millivolts. So it becomes really positive when you're changing the movement of sodium. Okay. Now I'll show you how sodium moves. Then that potential drops. Okay. The membrane potential drops. This is in volts, now millivolts. It drops down. It becomes hypopolarized. It gets almost down to the potassium voltage down there at minus 75, where it's pure potassium moving through the membrane. Thus, you have that action potential, and that mapped onto the membrane potential of just that pure ion. And then it comes back to the resting state here, which is at minus 61 again. So this is the actual movement of an action potential through a neuronal synapse, okay, through how you signal transduce through neurons, right? Very basic stuff, action potentials. So the transmission of an action potential is much more complicated, you might guess, because everything in biology is more complicated than even in things like chemistry, because you have biology and then you have chemistry. So you have, a sodium channel where it's closed and inactivated, right? So the sodium can't move in. So the only place it can go is inside the membrane. It can't move through the membrane, right? The only thing that's moving through is that potassium, and that's your resting channel. It's a resting state. So this is a closed system, and this is along the fine axial gradient of the axon. Now, when you move or when you activate that, okay, you go from this membrane potential where you're down here again at minus 61. There's a refractory zone that's displayed here. And then when the action potential opens up and that sodium starts moving through, that's when you get that polarization shift. Okay, then you get a depolarization back down as you're moving along the axon to get back to the resting state. After this dip, there's an increase, and then there is a regular steady state. Okay, so that's how you can measure it. Now that moves along the axon. See how this is moving? It's moving along vectorially from just showing from left to right, because the ions are moving along this membrane axis. This membrane axis is positively charged on the outside, right, and negatively charged on the inside. So as ions move through, they move through on a concentration gradient that's based on the charge distribution of the two ions involved. So that's all it really is when we talk about action potentials. Now, there's other ions involved, such as divalent cations like calcium, which make it a much more complicated dynamic. But for right now, I just want you to understand the basic action potential, because now I want to talk about myelin, okay, which is right now. So positive charges exist on the outside of the axon and negative charges on the inside. That's typically how it is. Shown here is like a node of Ranvir, we call these, named after the scientist that measured this in myelinated axons, okay? His name was Ranvir. So consider the consequences of delivering some stimulus to the point of the middle of the axon. If the depolarization is sufficiently large, voltage-dependent sodium channels will be opened, okay, will be open. That's what's happening here. And an action potential will be initiated across where this myelin works. So these are open parts of the myelinated sheath. So the myelinated sheath has some myelin and some unmyelinated track. It's still the white track, right? It's still the white matter track, because all this myelin comes off as white colored because it's a lipid. It's sphingolipid, sphingomyelin, for example, or galactosylceramide, compounds like that. So what you get then is what's called saltation. You get a hard charge distribution in the node of Ranvir. Then you get a rapid flux, because this is an insulated axon, of that action potential to the next node of Ranvir, where more sodium can move in and onward. And that's just showing you how that entire action potential moves down the gradient. Now, the only difference between that and the one I just showed you is that this is a myelinated axon. So that myelination, or that insulation that the lipid layer allows, this myelin sheath is a lipid layer, I told you, sphingolipid. It doesn't allow the electrons to stray. So you keep a very strong electrochemical potential without losing any of that potential as you move down the axis of the axon, okay? Down from left to right, shown on this particular slide. So then the positive charge will move to the adjacent region of the membrane. That's what's going on here. As the charge moves, it will depolarize. We saw the action potential depolarize. If it depolarizes sufficiently, remember, it has to go all the way down, voltage-dependent sodium channels in the adjacent region of the membrane will then become opened up, and a new action potential, new in quotation marks, it's not really new, it's the same action potential, it's just being transferred downstream because of this movement of saltation of that electrochemical gradient. So the charge distribution will then spread to the next region, initiate, as we say, a new action potential. It's really not new, it's the same one, it's been moved physically, right? So it's a geographical movement, if you like that term. I like that term. All right, so just to finish this up, propagation of the action potential in the myelinated fiber, which is what you're looking at here, starts with an action potential at the node on the left, okay? That's this one here, okay? In the absence of myelin, there's no myelin here, see? The action potential would propagate actively, normally, through the simple mechanisms we already talked about. However, now the myelin occludes the voltage-dependent sodium channel, so the action potential cannot propagate actively, so it has to move within the membrane, right? That maintains the activation and it speeds it up. It increases the rate. That's why axons are so important for nerve transmission. That's why we have higher thoughts. That's why you can understand the communication I'm relating to you in the English language, because it's much more sophisticated than something like a motor neuron, where you get an action potential and you get a change in electrochemical potential that can ultimately be related to a motor neuron innervating what? A myocyte. So you get muscle contraction, right? In the brain, the central nervous system, to get really high level signal cascades along an action potential, it has to be a lot faster. Otherwise, you wouldn't be getting when I'm talking to you at the rate I'm saying it, but you would be hearing it like this, or slow it way down. You wouldn't even know that I was saying words. You wouldn't even hear the letters. You wouldn't hear the syntax. You would hear nothing. You would hear just like beep, 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 right? So this saltation process increases it so fast, it starts to turn into communication, which is rarefied and far more complicated. So you can get a nuanced communication getting a myelinated sheath. It actually gives you a lot more information because it moves a lot faster along this axon. So that's really what I want you to understand why white tracks are so important. Sphingolipids are so important. There's a lot of leukodystrophies and a lot of sphingolipidoses, which are inborn errors of metabolism, diseases of sphingolipids, which cause a lot of neuropsychiatric conditions and a lot of mental illnesses that are inborn, right? Some severe cases, actually, which I've talked about also in the past. All right, so a brief thing about myelin sheaths and multiple sclerosis. Excuse me, I have this cough. Myelin sheaths are interrupted with nodes of Ranvir, which you just saw. Here, the axon is in contact with the extracellular medium. So it's a raw, if you want to consider it, a raw conducting axon. Sodium channels are located at high density there, right? And at that unmyelinated region, the action potential saltates. Saltare is Latin, that means to jump or to move between nodes. Actually, it was first described for like sand particles moving along on a beach, for example. So nerve transmission obtains due to an ionic current via ultimately a passive conductance of these ions. This is faster than the continuous propagation of an action potential, but is also open to rapid dissipation, kind of like how electrons move through an uninsulated copper wire. That's important too, though. You want to be able to know what I'm saying, and then you want to be able to hear other sounds in the room. For example, the lion in the corner, right? Or the person knocking on the door. So there are a lot of different things you have to process in your environment, not just what's inside your head, not just the reasoning powers that you have in your central nervous system and all of the possible cascades and signal transduction events in the brain, in the central nervous system, and then leading out to the rest of the homeostatic system in the body. You also have to be able to work in real time when something changes or happens, like me talking and then me stopping. Okay, so the faster the continuous of the propagation of the action potential, but it's also open to rapid dissipation. I said that already. Therefore, the nodes amplify the signal to maintain the intensity of the electrical stimulation as if it was at the get-go. The myelin sheath creates that environment for that to occur. Without that myelin sheath, the electrical impulse becomes attenuated, ion leakage can occur, and the impulse doesn't capacitate to the next node. And that's what happens in multiple sclerosis, because there's a demyelination of the axons. That's what multiple sclerosis is. It's a terrible disease. I had a student once get that disease when I was teaching at the University of Colorado, and it was just a terrible thing. All right, now we're going to get to the paper. Paper is white matter microstructural differences across major depressive disorder, bipolar disorder, and schizophrenia. A track-based spatial statistics study. And that was published actually, it's going to come out. So this is a pre-publication I can read because I have access to the library. It's in the Journal of Affective Disorders, volume 260, and it's going to come out in January of 2020. So you'll have to wait for this for a couple of months, but we're going to talk about it today. Here, it's only October. And those are the page numbers you'll find relevant. This is actually a photo I took with my camera on my phone one morning when I was walking. I walk every day along this arrow ridge here, and my home is back over on this side here. And that is indeed, ladies and gentlemen, the Clearwater River, right, which drains the western continent. One of them leads into the Snake, and the Snake then into the Columbia and onto the Pacific Ocean. In fact, this is where Lewis and Clark came down, right down that river. I know you wanted to know that. All right, so let's introduce this paper. Clinically distinct major depressive disorder, bipolar, and schizophrenia do not obtain clearly separated diagnostic categories, even in the in the fifth version, right, of the statistical manual. As patients share those clinical characteristics such as mood swings, perception changes, and cognitive deficits, sometimes motor deficits too. The endophenotypic neurocorrelates, therefore, offer some unique and maybe convergent transcriptome signatures, which means you might see some unique fresh transcription of genes, that is the production of RNA from the DNA, transcription, beginning of gene expression, right? But you also get a lot of overlap, so convergent transcriptomes. Moreover, neuroimaging reveals common structural neuroanatomical and therefore functional neurochemical molecular patterns that map to a loss of gray matter of, for example, the anterior insula and the dorsal anterior cingulate via measuring it with a voxel-based as well as a connectivity MRI type of system like we were just looking at. So structural connectivity suggests, okay, this is the background for you to take this paper as a way to look at the neuroendophenotype of a neuropsychiatric disease, right? So you have to take this whole amount of premise or proposition in order to be able to come to the conclusion that what you're looking at with the scan actually maps to disease. Okay, so these are nothing more than premises or propositions to the argument. Structural connectivity suggests, always look at that word there, functional networking, where psychological presentation of these major diseases obtain fiber tract connectivity disruptions. I can imagine disruptions in that node of Ranvir plane of both the afferent and efferent connections, those neuronal connections between regional neocortical areas. And we'll talk about which ones those are soon. So those areas are associated with initiation and subsequent pruning back of axonal connections where diffusion magnetic resonance imaging, that's small d MRI, has revealed alterations of white matter integrity, is the term they use, and coherence of measuring what's called fractional anisotropy. Okay, so differences in structure, therefore, should map onto differences in function, you see, because of anisotropy. Of course you do. So there's something, I'm sorry for these acronyms, but this is how it's done in the field. Tract-based spatial statistics, TBSS, provides a detection of the fiber tract change across the brain. It's actually across the membrane too, as is implemented to provide voxel-based. Okay, so now you can start to do quantification because you use voxels, right? So you have a density of space, and that's what the voxel is going to measure. Evidence of a structural disconnectivity. Sorry, I don't know if that's a real word. I kind of made it up. In MDD, BD, and schizophrenia, demonstrating aberrant white matter integrity. Okay. In fiber tracts, such as here's I told you I would tell you which regions, corpus callosum, the corona radiata, revealing shared white matter abnormalities between those three diseases. Okay, either between MDD and bipolar disorder or between schizophrenia and bipolar disorder as compared to healthy controls. Okay, there's a reason why we're grouping them this way. You'll see in a moment. So the data showed, this is what they're saying, there is convergent and divergent white matter microstructural change, okay, across those diseases using that spatial statistics program to look at the anisotropy that's measured by the DMRI, okay? So it's hypothesized that patients with major depressive disorder, bipolar, or schizophrenia would show a diagnostic across all those diseases pattern of white matter alterations in the corpus callosum and the corona radiata compared to healthy controls. So here's what they did. Patients with MDD, they had 85 of them, BD 42, so about half, schizophrenia somewhere in between, from second affiliated hospital at Dijon Medical University that were included in this study over the 4-year period between March of 2013 and October 2017. So this is a Chinese study. These diseases were confirmed by psychiatrists using the structured clinical interview for DSM-4 TR. The exclusion criterion included a diagnosis of any other mental illness, organic causes of depression, for example, heart disease, liver disease, or kidney disease. Now, again, when you do an exclusion criterion, I mentioned this last time in Authentic Biochemistry, you're actually not getting a cross-section of the population. Who walks into an office that has a mental disorder? A cross-section, not a cohort. So whenever we do cohort studies, we do that to control the variable, control the variation, so that we can look at just MDD, just BD, just schizophrenia. See what I'm saying? Those would be some of the markers because these are the diseases that have been assigned to these people, right? Because they've taken the correct scheduled questionnaire, right? And they say, yeah, these people have MDD, these people have BD. See what I'm saying? But they don't want people to have any other confounding diseases. But you know that people have confounding diseases. And those are things that often happen when somebody shows up in a psychiatrist or psychologist's office. And if you're not thinking about that, you're going to have a lot of variation in each individual person. And that's really what I get to when I'm trying to describe all of biomedicine. Each individual, because of epigenetic phenomena, because of the immunoepigenome constantly interacting between the genome and the environment, something I call dia-event ontological perspective or paradigm. Because of all of that, each individual is actually unique. So when you do these kinds of studies, remember we're grouping together to try to decrease variation around the mean so that you can get statistical changes and you can say, aha, this relates to this disease versus that disease. See? So it's a built-in bias. And that's basically in all of science, not just biomedical. Plant science, microbial science, you name it. Just so you know. Okay, the Beck Depression Scale, BDI standard, and the Beck Anxiety Scale, BAI, are used to assess symptoms in patients with MDD and bipolar. The scale of Young Mania Rating Scale, the YMRS, is used to assess the symptoms in separating out the bipolar people. The bipolar patients further diagnosed into mania and in the subtypes. So you can see there's a spread there too. So here's a paper from Psychiatry Research in 2012. Now we're going to go back here. Why am I doing this? That's what you do when you want to look at a paper that's published now. You want to take the time and read it very carefully. You want to see how the experiments were done. You want to see what the data looks like. You want to see if the data rises to the level of evidence. And then you want to know how the evidence is described by the authors by looking at the evidence, looking at the data as it's presented in their manuscript. It's published. These are refereed papers. The only thing I do here at Authentic Biochemistry and VerEvMed Studios are published papers that have gone through peer review in real journals, okay? Not anything that's popular press, newspaper articles, or it's just someone describing something. I'm going directly to the papers. That's what I do with VerEvMed and with Authentic Biochemistry. That's why we're unique. Okay, so back to this. This paper published several years ago, I had to go back to so you know more about this whole white matter track analysis. And this one was looking at reduced white matter fractional anisotropy, called FA, and clinical symptoms in schizophrenia. So this was when it was first described in schizophrenia. Voxel-based DTI, which just simply means diffusion tensor imaging. It's a way of doing your MRI. It's a way of tuning your MRI. Now, why did I go back to 2012? Because that was the first time it was published. So I want to tell you if the paper we're looking at 2020, which come out in January, right, is coming from, where's it coming from? Where do we get this kind of analysis? This is where schizophrenia was first described using FA and DTI. What does it say? Although not consistently replicated, important line there, DTI studies in schizophrenia have revealed lower fractional anisotropies. You get a lower value in various white matter regions, something we're talking about, remember those myelinated areas, a finding consistent with disruption of white matter integrity. So they're saying that they get this DTI with that kind of imaging. They say that because of that, it translates to a lower FA, fractional anisotropy, okay? And that then looks consistent with actually disrupting the integrity of the white matter. Okay, that's what this paper established in this publication. Okay. Compared with the controls, let's say it's not consistently replicated though. Very important. Compared with the controls, the schizophrenia patients showed significant FA reductions, those are fractional anisotropy reductions, in bilateral superior longitudinal fasciculus, bilateral inferior fronto-occipital fasciculus, and the genu of right internal capsule. Okay, so those regions, those nuclei of the brain. Okay, sorry, but stumbling over that. It's fasciculus. Fasciculus is how you pronounce that. So the FA, remember that's the anisotropy, value of the anterior part of the corpus callosum, remember that's one of the regions we're looking at, the CC in the 2020 paper, was negatively correlated with the avolition score of the scale of the assessment of negative symptoms. So that means it's linked with the assessment of negative symptoms of the disease. What disease? Schizophrenia. Those findings, then, they say in this paper, this seminal paper, if you want to use that term, suggest widespread disruption of white matter integrity in schizophrenia, which could partially explain or partly explain the severity of negative symptomology. Now looking at positive symptoms, looking at negative symptoms, which makes sense. So here we're looking at the actual data. The overlapping regions with consistently reduced fractional anisotropy for major depressive disorder, bipolar, and schizophrenia compared with healthy controls. And you're looking at corpus callosum and the corona radiata from left. Okay, so here is the body of the corpus callosum, and we're looking at now MDD, BD, schizophrenia, healthy control. You can see that these are all depressed. Here's the convergence, the lower FA for all three neuropsychiatric conditions as compared to healthy control. The GENU of the corpus callosum, same thing. They are not different amongst each other statistically, but they differ according to, statistically differ, that's what those little asterisks are, for some of the domains of either the three diseases in the healthy control. Then you look at the anterior radiata, and you see much lower, this is interesting, right? Lower fractional anisotropy compared to the corpus callosum in general. So that tissue is going to give you a lower signal at the get-go. But notice again, statistical differences with the healthy control and statistical differences on the right anterior, right, of the corona radiata. Here you get a really strong difference between the left posterior of the corona radiata, all three diseases, again, converging against the healthy control. Same thing with the right posterior, so they look very similar. And then finally, the right superior corona radiata, you can see that some of these are very subtle differences. But you can see that MDD falls out here. MDD is much lower than is bipolar, than is schizophrenia, which are much closer to the healthy control. So this they're now seeing divergent and convergent changes in the measurement they call fractional anisotropy, which is a measure of the disruption. If you believe the earlier papers, okay, and you have no reason to discount it, but you have to know that you're carrying that forward when you look at this data. This data, again, is from the 2020 paper. All right, cool. So here is some really cool scans. So let's move me out of the way here. So the anisotropy differences between MDD and schizophrenic groups. Okay, this is showing you the scans themselves. Anterior portion, okay, of the anterior CR, remember that's the radiata, showing significantly reduced anisotropy in MDD patients compared to schizophrenic. So that's the red, okay? So that's the red. You see a great reduction in fractional anisotropy compared to the schizophrenics, which are in the yellow. See, they're right up there, with healthy controls on this measurement, right? Okay, continuing, the posterior portion of the anterior C showing consistently reduced anisotropy for all three, MDD, BD, and SC, patients compared with healthy controls. That's the blue, right? So you can see that there's much less difference here between schizophrenia and bipolar with the healthy control, okay? In the left anterior radiata, corona radiata, as compared to the right anterior, where you see that there's, again, a differential, right? So what this says is compared to the schizophrenic group, the MDD group has significantly reduced FA measurements. This is what you're showing you here, that red region there, you see. In the bilateral, mapped here on the histogram, in the bilateral anterior portion of the anterior corona radiata. Okay? In contrast, the overlapping region with the decreased FA across MDD, BD, and SC groups was mainly in the posterior portion of the anterior, right, corona radiata. So there are now what? There are structural domains within the central nervous system that appear to have some convergence of the FA, the decrease in the anisotropy for all the diseases. And then there is some divergence related to specifically major depressive disorder. These are from patients. These are scans from patients, and these are quantification of those patients. Remember, from that Chinese cohort. Now, remember the number of patients, right? There's 1.3 billion people in China. How many of them suffer from neuropsychiatric disease is probably anybody's guess, but maybe up towards 20% of the adults, maybe more. So how much would 20% be of that 1.3 billion? Well, that would be hundreds of millions of people. We're looking at 140 people. So this is a very, very small N number, right? So that means that, is this going to map out if you looked at 500,000 or 1 million people? No way of knowing if the statistics would show up. Always remember that too when you read a paper. Even though it's a published paper, I'm not putting it down, this particular paper. I'm just giving you a clear idea of what a sharp scientific perspective is when I read the scientific literature. You have to be skeptical. It's a brand new paper, and they're trying to tell you by doing this DMRI, right, and using the FA of that, using DTI, they're saying we scan some of our patients, we can say, look at that white track anisotropy. That's MDD. See, and we have the person. They've taken the questionnaire, right? They've used the instrument, and you say, they're depressed, and they got this white track change. But remember, N number, very small. And remember, we're suggesting that these structural changes, which is all this is, maps onto functional changes. And we're saying there's a mechanism by which this occurs. In the second paper, we'll talk about that, the whole lipid dyslipidemia pathway. Okay. You're going to love that. All right. So let's analyze the data from here. There was a consistent transdiagnostic pattern of white matter microstructural alterations in the body and genu of the corpus callosum, and bilateral anterior, posterior, and right superior corona radiata across patients with all three diseases. There's consistent transdiagnosis. A gradient, however, in the common alterations was observed in the anisotropy, with MDD being less than BD versus schizophrenia. And that's what we saw. Remember, schizophrenia being much closer, which is really interesting, to healthy control for this particular parameter, right? In contrast to the shared structural disconnectivity, therefore, diagnosis-specific effects were only found, they say, but only one that fell out was MDD. And that was only in the anterior portion of the anterior corona radiata. So what's my dialectic here? Remember what a dialectic does? It says, what is our first principle and how do we reason towards it? Okay, so we have to come up with premises, and we have to see if those premises are true. And if they're true, do they reach a conclusion? And therefore we have a sound argument, right? So, and that's just a fraction of what the dialectic does. Basically, dialectic tries to prove or to try to gain truth by finding it through reason, but increasing the amount of empirical input as you do that. And that's what the analysis is. Empirical input here is what? The data. The reasoning is the whole thought process involved in designing the experiment, right? Which has to do with the faculty of the understanding, which has to do with conceptualization and schematization of those concepts towards ideas that the faculty of the imagination creates, so that you can come up with a hypothesis or hypothesis to generate experimental design, to do this kind of experiment, get the data, turn it into evidence, and discuss it. Yeah. All right. Now, what they're saying is the anisotropy is a reliable scanning diagnostic tool for the indication of MDD. However, it may not be specific. That's one of their major conclusions. Kind of weak in a way, right? But it's honest. White matter fractional anisotropy differences in the corpus callosum are consistently reported in those three subtypes of neuropsychiatric disease, where reduced anisotropy in the corpus callosum showed greatest effects, usually in schizophrenia, in a very large-scale prospective meta-analysis study. So they were looking, so that's a lot more people. I don't remember how many were in it, but thousands only, sorry. The corpus callosum has the largest white matter tract, connects the bilateral cerebral hemisphere, the brain anatomy, and enables communication between those hemispheres. So it's a really important part of the brain, right? It connects the two hemispheres. The genu of the corpus callosum in particular connects the prefrontal cortex and was also identified as the shared reduced fractional anisotropy between the bipolar disorder and schizophrenia, and between bipolar disorder and MDD, major depressive disorder. So, aberrant white matter microstructure in genu and body of the corpus callosum suggests abnormal interhemispheric, but I pull this back up here. People complain when I'm half cut off there. Suggests abnormal interhemispheric information that's between the two hemispheres may play a role in the pathogenesis of all three neuropsychiatric diseases, major depressive, bipolar, and schizophrenia. Combined transdiagnostic evidence therefore suggests there is a common structural substrate that is the actual what? The nuclei of the brain, the central nervous system, of interhemispheric connectivity of the white matter tracts across major mental disorders. So how do I conclude from that? That was the dialectic. Now let's do some conclusions. So the corona radiata is embedded within the pathway within the anterior insula. It has been identified as a region associated with gray matter loss. This has already been described across many mental illnesses. The CR is with emotional and executive dysfunction. Okay, it presents that way. And the anisotropy reductions in the corona radiata were detected in the 3 diseases we looked at, with the DMRI measures in the radiata associating with speed of information. Now they're telling you that they think that it's associated with the speed of information. It has to do with exaltation, right? White matter tracks, processing and working memory performance in patients with mental disorders. That's what they're arguing here. Specifically, the anterior corona radiata, which contains reciprocal connections between the anterior thalamus. Thalamus plays a major role in a lot of neuropsychiatric disease, I don't know if you know that. And prefrontal cortex plays a role in neural or neuronal circuitry of emotion regulation, so that affect portion of major depressive disorder, bipolar disorder, schizophrenia et al., has the correlation to MDD presentation criterion. That's what they're saying here, okay? They're trying to say, you look at the structural changes on different regions, nuclei of the brain. You're now saying there are functional changes. You're already arguing that point of view because how the white matter tract works, although you haven't shown a mechanism here at all. And then they're saying, finally, that endophenotype maps onto the presentation you see in the psychological state or the psychiatric condition now. In contrast to the convergent white matter integrity abnormalities in the posterior portion of the anterior of the CR, the MDD-specific pattern in the bilateral anterior portion of that region, of that nucleus, of that anterior CR, corona radiata, suggests that the anterior portion of the anterior corona radiata is a potential biomarker for the identification of MDD. That was the whole purpose for the paper. Okay? So there you go, right? So that was a 2020 paper, and I just wanted you to get an idea of what we're doing now with neuroimaging, okay? So I want to do a little bit of just discussion of just major depressive disorder, and then I'm going to go ahead and publish this video lecture, and then when we come back, I'll come back to general description of some of these neuropsychiatric diseases. And then I'm going to jump into paper number 2, which has to do with leptin, the adipokine leptin, and how it may have an effect on white tract anisotropy. There you go. So I found 2 papers I thought you as my audience would like to get this weekend. Okay, so let's just go very basically through this. This is from the Neuropsychiatric Disease Treatment paper published in 2016. Pages included there. Major depressive disorder is a leading neuropsychiatric disorder which is predominantly mapped onto the monoamine neurochemistry. So now we're getting more into what I feel more comfortable in, biochemistry, neurobiochemistry, right? There we were doing basically neurocellular physiology or neuronuclear physiology, right? And using MRI, which is something I've forced myself to learn as well, of course. But all right, so diagnosed in both sexes, MDD, and many age groups, but most common in young or early middle-aged adults. In the U.S., up to about 16 million people, that's about maybe 7% of adults over 18, report at least one major depressive episode in the past year, usually because of some terrible episode that occurred, depressive episode following an episode like the death of a family member or something like that, or some kind of major trauma like a car accident. Most prescribe selective serotonin reuptake inhibitors. We all have heard of these, SSRIs. And the selective serotonin norepinephrine reuptake inhibitors are also being used. And there's a reason why you might use one over the other. One of them has a broader range, and because norepinephrine and serotonin sometimes co-migrate to similar synapses, and so if you can cover both of those neurotransmitters, serotonin and norepinephrine, that is, you might be able to better treat some forms of MDD, unipolar MDD in particular. And the study, that study, which examined adherence to prescription, so that one did desvenlafaxine, which is an SNRI, was adhered to more than the SSRI, SNRI drugs that they looked at. Okay, so this paper was actually very specific, very clinical, and I've covered this in the past VerEvMed lecture. And really, the reason I wanted to look at that is because are SSRIs and SNRIs doing their duty? Are they really relieving the terrible condition of depression in the American population or in the human population in total globally? And as it turns out, maybe not so. So we're doing now more fine-tuning of the chemistry so that we can figure out different ways of agonizing or reverse antagonizing some of the receptors for some of those major neurotransmitters to see if we can modulate more carefully that neural transmission and see whether or not that can play a role in giving some relief to some of these neuropsychiatric disorders. So what I'm also talked about, and I will a little bit when we come back, is there is an alternative to the monoamine. These are called monoamines because they have one amino group in these compounds. And it focuses on the glutamatergic neurotransmitter system. So we're moving now, shifting a little bit from 2016 up to 2019, 2020, not just talking about serotonin, right? Not just talking about norepinephrine, which are monoamines, but talking about polyamines, such as the glutamatergic pathways, as potential targets using high definition, high profile, heavily targeted specific neurochemistry to alter and to therefore possibly relieve major depressive disorder. So what we want to look eventually is that larger picture, obviously, and that's what that paper does. Okay. So I'm going to stop here, close that off there, put me over there, and come back down to here. I'm going to stop here, and when we come back, what we're going to do is talk a little bit more generally about some of these neuropsychiatric disorders. Then we're going to jump into paper number 2, which is really fascinating. Again, a brand new paper this time was published in 2019. And that paper is going to describe, again, we're not doing pharmacology here. We want to know about physiological and specific diseases that could be associated with neuropsychiatric states and conditions. And one of those diseases that is prevalent, as we all know, worldwide now, is dyslipidemia as it's related to obesity. So does obesity play a role in neuropsychiatric presentation? And indeed, does it play a role in the conditions that favor the etiology and even the induction of the etiology of those neuropsychiatric diseases? And that's what I'm going to do next time. So we'll have much more fun, I think, because there'll be more synthesis and a lot more biochemistry. Unfortunately, not too much lipid biochemistry, but don't worry. Lipid biochemistry, we're always going to get back to it eventually. But anyways, we're going to stop there. So now I'm going to say, again, on this date, the 19th of October, 2019, you can see what time it is, Pacific Standard Time. This is Dr. Daniel J. Guerra from VerEvMed, an Authentic Biochemistry Studio, saying bye for now.